So, good morning for everyone. I'm very happy to deliver our lecture on this great AOPT meeting, and we are sending our greetings from Finland, from Kuopio. We are in the middle of Finland, in a small, very lovely city. In a University of Eastern Finland, I am working with a great team with Professor Kai Niranta and with his teammates. And today I would like to talk to you about the role of NRF2 and PGC1 in oxidative stress. More closer, we will show you data on epithelial to mesenchymal transition. But first of all, please extend our acknowledgements to Professor Hepping. Thank you for his great invitations and collaborations, as well to my boss, Professor Kai Niranta, who always give new ideas to be in action. Ali Koskela, great collaborator from our group, Mei Chen from Belfast, and a Polish professor, Professor Blasiak, for his great achievements in this project. As you can see, we are a cross-border team. You know, Finland takes the most coffee in the world, but why it's problematic? Because we love to make coffee and to take it. Our max, it's our germs, but if and whenever AMD starts to develop, we have problem to recognize the signs on our max. Or if it continues without treatment, that most of the patients lose gradually their central visions, and we cannot recognize our max. It's a real problem. So some facts about AMD. As you know, it's a burden problems all over the world, having 196 million of patients and billions of dollars, euro, or even yens taken for these diseases. As you know, AMD affects eye, more closer the retina. And if you see on the schematics, you can see that AMD affects photoreceptors and retinal pigment epithelial cells. On this immunohistochemistry from human, you can see that some of the stress markers are highly expressed in AMD cases, like ubiquitin and PC62 increase dramatically compared to the healthy ones. Although the general role of AMD in AMD, especially in wet form, seems to be well established, the mechanism of AMD in dry AMD is less known. So therefore we decided to investigate these questions in our model. To study AMT, its underlying mechanism, we used a transgenic mouse animal model. We generated mice having knockout for two genes, NFA2L and PGC1 alpha genes. This study has been published earlier, so we know that this novel mouse animal model is having AMD phenotype, and it has been published a few years ago in Redox Biology with the international team. In our current work, first we set out to elucidate how NFL2 and PGC1 alpha affects RVA morphology at light microscopy level. As you can see on the upper two image, low magnification from the mouse retina shown, and the box region is highlighted, showing that there are more RPA cells in wild type. And we have quantitated the data, and as it has shown, there are significantly more cells have been recognized. Next, in order to test whether the discovered change in the nuclear linear packing density reflects altered cellular density and/or arrangement, we made phalloid flat mount experiments. And as you can see on these two images, the yellow arrowheads indicates alteration in the phalloid pattern in the DKO. Also, the wild type show normal hexagonal typical cells in contrast to DKOs, where the cellular density varies somehow, and also they have lower cellular density. as you may recognize on the lower image. As alteration of cell-cell adhesion may promote cellular phenotype changes, we next used TEM to examine the cell-cell contract in the RPA layer to further understand the possible role of these genes. As you can see on these TEM images, normally developed tight junction in wild type, in contrast to the DKO, in which irregularly shaped apical tight junction has been recognized, suggesting a breakdown of outer barrier, and clearly suggests that the loss of these genes affects cell-cell junctional complexes. Next, to answer the questions whether the loss of these genes induce AMT-like changes, we looked for the structural alterations in cellular architectures. And as you can see on these images, in DKO model, the RPA lost their basal infoldings and also observed elongated basal laminar deposits and thickened basement membrane, in contrast to the normally developed wild-type AM. As you can see on these TEM images, we were also interested in the apical compartment. On the wild-type, we seen densely well-arranged microvilli and tight contact to the photoreceptors, in contrast to the DKO, where the loss of this chain strongly affected the arrangement of this system. We also were interested how the height of these cells alter at ultrastructural level. And as you can see on this graph, the height of RPA was markedly decreased upon the loss of these genes. Next, certain AMT marker has been analyzed using confocal microscopy, and if you see the lower row, there were altered expression of these markers for this model. And we did semi-quantitative analysis for this, and you can see the statistical differences on each particular graph. As a summary, TEM and immunohistochemistry analysis shown here suggest that the loss of these two genes in mouse RPA cells in vivo and in vitro triggered morphological, mesenchymal, like ultrastructural and light microscopical changes. As a summary, average lateral dimension of some of the DKO RPA cells increased, notwithstanding the relative decrease in their thickness, as it illustrated on this schematic. Such morphological changes in the average of cell volume might be indicative of mechanisms whereby DKO RPA cells are able to balance their flattering rate in order to cover defined tissue size and to try to maintain their potential role in supporting photoreceptors' function. Detection of lipofucsin is one of the feasible methods used for identifications of senescent cells. And as you can see on these confocal images, lipofucsin was highly expressed in the DKO, and on these TEM images you can see the increased number of lipofucsin granules we analyzed. And we made accurate statistical analysis, as it's shown on these two figures, which suggest and demonstrate that lipofucsin accumulation positively correlates with the common loss of these two genes. Finally, we were interested about senescence markers, so therefore we did confocal microscopical analysis, and as you can see on this immunohistochemistry and also on the quantitative analysis on the bars, that we and others have shown also that senescence could be an important factor in the AMD pathogenesis. As you can see on these schematics, where simplified representation of the oxidative stress-dependent major molecular mechanism of epithelial mesenchymal transition is shown, we labeled what its happening in our animal models. Our results also suggest that the dysregulated response to oxidative stress is critically involved in fibrosis. But also we think that oxidative stress is involved in various proliferative retinal diseases such as NMED, PDR and PVR. Oxidative stress mediated EMT may also play a critical role in those conditions. At the end, I would like to thank you for your attention. I was really amazed on different videos of these conferences, and I would like to thank you for your attentions. As I am Hungarian, köszönöm. If you are interested to know more about University of Eastern Finland, visit the website. If you are interested to know more about Professor Karniranta AMT team, please follow us on Twitter or visit us on our own website, karnirantalab.fi. Thank you again, and I am ready for your question.